Hello, everyone, and welcome to today's Keynote webinar. Excuse me, sorry. Welcome to today's Keynote webinar titled Timing is Everything, Deconstructing the Ebola Virus Infection Program by CRISPR with Live Q&A. This webinar is a part of the 11th Annual Microbiology Virtual Event Series. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. Okay, now let's go ahead and get started. I now present today's speaker, Robert Davey, PhD, Director, Professor, National Emerging Infectious Diseases Laboratories, Boston University, Boston, Massachusetts. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Robert, you may now begin your presentation.
Thank you, Sydney, and thank you for inviting me to the symposium. I think it's a great opportunity to talk about my work and also to engage with people out there who are interested in what we're doing. So, as Sydney mentioned, the title of the talk is Timing is Everything, Deconstructing the Ebola Virus Infection Program by CRISPR. So what's this about? So the viruses, like other infectious agents, are following a pre-programmed set of instructions of how to get into the body, alter processes that are ongoing in cells and the body, to set up shop, to create more viruses, to suppress the immune response, and then to spread the infection to other people. So the goal of my lab is to try and understand this program, the timing of the program, and to intervene and disrupt the program in such a way that the virus, in the case we're talking about here today, Ebola virus, the infection is disrupted and it can no longer proceed and spread from person to person. Trying to think about therapy as the ultimate goal. So I just want to, this is the cover of Time from 2017. And the quote from the magazine in 2017, just read it here, is, "For all the advances in finding dangerous pathogens," so in other words, detecting them, "the simple truth is that neither the world as a whole nor the United States in particular is at all prepared to handle a major infectious disease outbreak. And a significant reason is to the lack of investment in how we control and detect and then treat infectious disease." And that was before the pandemic, the COVID pandemic happened, and we learned hard lessons during the pandemic. So the NEIDL, which I'm directing, is a high containment laboratory that's in Boston. It's part of Boston University, and that's shown in the picture at the left there. And it's a high containment lab, which means it's the highest level of containment, where you work in these person in the bubble type suits, and the suits are connected to air lines which come down from the ceiling, shown as these red coils, and you enter the lab. The suit inflates, And if you get a puncture or something in the suit, air blows out through the holes and prevents the viruses getting to you. And it's a very, very safe system that was actually developed in the United States, and is the sort of state of the art in high containment safety work with highly pathogenic organisms like Ebola virus. And what we do in the lab is we work on antiviral therapies. A lot of it is drug development. We also do vaccines. And we try to study how the virus replicates in cells so we can intervene and disrupt the infection process towards therapy again. And we do everything from basic testing of vaccines and small molecules all the way through to preclinical and then even regulated studies to get things through to licensure and through the federal authorities in terms of getting things to the clinic. So my lab in particular, what we like to do is we like to work on 3 main viruses: filoviruses, which Ebola virus is one type; arenaviruses, which Lassa fever virus is one type; and another virus called Crimean Congo hemorrhagic fever virus, which is a tick-borne virus spread by ticks. And it's there's not a lot of known about this particular virus, but most of the work that we do is on Ebola virus and Marburg and its related virus, Marburg virus. We the icon in the middle with the hand holding up the pills there is meant to represent our main focus of interest, which is trying to identify small molecule therapies which disrupt protein-protein interactions, shown by the blue and the purple sort of blobs in the middle there. And the blue blob is meant to represent a virus protein that interacts with a host protein, and when these two things come together, that establishes the disease state or allows the virus to replicate in the cell. If you disrupt the interaction of the two proteins through a small molecule drug, you can prevent infection and stop ongoing infection. The icon on the right there showing the cell, we like to also try to identify host proteins, namely the purple blob there, by knocking out host genes or mutating them in some way, such that we can identify those host proteins that the virus relies on to set up shop inside the cell and to circumvent the immune response, and go into sort of stealth mode to replicate itself. So if we identify such proteins by, say, removing them from the cell, we then expect to see the virus can no longer replicate in the cell, and therefore we can try and target that particular protein inside the cell and stop virus replication. And all that is sort of exemplified by a paper we published in 2021 in Proceedings of the National Academy of Sciences, where we're looking at what's called the overlap of the viral interactome, which is indicated as that virus-like object on the left of the middle of the screen, how that interacts with the host interactome, which are those green circles indicated as a network on the right, and the overlap between the two things is called the virus disease module, and that's a network of host proteins that the virus is directly interacting with. And if you disrupt this network in any way, you can stop virus infection. And the goal of my lab is to try to identify this network that's in the middle there, the disease module, and that's thought to be the thing that that is most easily attacked, and we can use that as a way of stopping infection. All right, so the big question is, how do we identify those things in the middle in that network? Now, a lot of people ask, how do you work? What type of workflow do you do at BSL-4? What is possible at BSL-4? So a lot of what we do at BSL-4, the lab is difficult to work in because you're wearing the bubble suit. you've got the airline attached to you. There's air blowing over your face all the time. So it's it's very dehydrating. And also, you're trying to make sure that you have enough, you've had your breakfast in the morning, so you're alert. So you can only spend a limited amount of time in the BSL-4 just because of the sort of biological limitations. So we try to limit what we do in there, and then treat our material. So what we do is, on the left there on that icon, is we do a lot of high throughput sort of preparation of materials. These could be, cells, antibodies, drugs, whatever, using simple liquid handling machines like the cartoon that's shown there, that fill up plates very quickly. And then you go into the BSL-4 and add the virus, as shown by these virus icons here, using basically the same type of machine. Fill up the plates with the virus, allow that to incubate for up to maybe, say, 36 hours or so the virus can replicate inside your cells. And then what we typically do is we treat that material in some way that's with chemicals, or for instance radiation, so the virus is destroyed and can no longer be infectious. That allows us then to take the material out of the BSL-4 lab and do more complicated processing of the samples, say staining with an antibody, or imaging with a confocal microscope, for instance, to really understand the infection process. So that's the type of workflow. So we try to spend as little time in the BSL-4 as possible, really, even though it's probably the most exciting part of what we do. Get it out and then process the material in the BSL-2 lab. And my lab is particularly good at doing microscopy imaging of infected cells. So the set of black and white images at the lower left there show infected cells as little dots. Each one of those little dots is an infected cell, untreated. And then if you treat with, say, a drug, you get less little dots. It's a relatively straightforward concept. We just count dots. And then we're also looking at how many total cells we have in each image, as shown by the cell nuclei down below. And you can see here that the cell nuclei are about the same in the left and right panels, but the little dots are less in terms of less infection, and therefore we can say that treatment is effective at blocking virus infection. And then we have some computational tools that we use. Cell Profiler is a wonderful open source software out of the Broad Research Institute in Boston, as well as Cell Analyst, that allow us to understand where the infection is disrupted on a, say, a 384 well plate, which is shown by those icons here of the different colored wells, that sort of thing. And we can get some nice quantitative data out of this process. All right, so that's the how, and that's the what, how, and how we do it. So the why of what we do. So Ebola virus, it's still out there. So many of you probably remember the outbreak in 2015. It was the world's largest Ebola virus outbreak ever seen in Africa. And the big deal was the virus went from where it's normally found in more central Africa, but got to the west coast of Africa. And from there, it migrated across the world. So there were people that got on planes that were feeling maybe not so well. But by the time they got off the plane, they were really quite unwell with full-blown Ebola virus infection. And this was a major concern for the world, mainly because of the fact that Ebola virus has a very high fatality rate, ranging from 40 to 80%. Some other facts about Ebola virus, which make it difficult to know how to predict what it's going to do next, is the natural host reservoir is unknown. It is likely bats. So there's some sort of reservoir, but recent information suggests that maybe even people can serve as a reservoir for Ebola virus. There was a couple of cases where people had seemingly recovered from Ebola virus, but they were able to infect others years later. So that's always a concern. How you know, and to understand what's going on there, you know, how does Ebola virus stay dormant for such a long period of time is still an active focus of research for many people. Fortunately, you know, there are some treatments available for Ebola virus. So monoclonal antibody therapy is available. I was involved in getting one of those to licensure through the US FDA. There is also a vaccine that works against Ebola virus, but this is usually used only in the case of outbreaks. It's not something that you and I would usually be given. It's only given when it's needed. But there's no approved small molecule therapy for Ebola virus. So these you know, the monoclonal antibody therapy works if you get treated early in infection. The vaccine obviously works if you're given it before infection. But small molecules, which could be used as an intervention after you're infected and after you're showing small signs of disease, there's no treatment available that's been approved for that, even though there are some candidates that are becoming available and being tested more thoroughly. All right, so that's, you know, it's still out there. But what I you know, today, you know, what I'm going to show you is more or less a platform that we can apply to many other viruses when they pop up. Ebola virus is just a sort of a poster child for how do we deal with emerging pathogens. You know, what is the best path forward in terms of developing a therapy? But what I'm going to show you shortly is, you know, we can apply these techniques to other virus types to rapidly advance towards therapies. All right, so two recent manuscripts that out of my lab that are good to read as follow-up to this seminar. One is the topic of today's seminar, which is a paper that was done by Rebecca Carlson from Paul Blaney's lab and Justin Patton from my lab. He's now at UCLA in the graduate school there, which is a fantastic outcome for Justin. They are joint first authors on a paper that was just published in Nature Microbiology, and that's single-cell image-based screens identify host regulators of Ebola virus infection dynamics. And this, I'll get into this a little in a short while. Another wonderful paper that we just published in Nature it's about to be published in Nature Communications is its protein proximity screen identifies Ebola virus host factors that are controlling infection in the cell. And we focus on a protein called EDC4 and its role in, and its part of the RNA decapping complex. But that's, I won't be talking about that today, but these two papers exemplify what we're trying to get at. Anyway, so back to the first paper. So this paper, what it's trying to do is it's trying to look at the infection program, right? The program that the virus is following to infect, suppress the immune response, and then to spread. And we use a special technique called optical pooled screening to do this, and I'll get into that shortly. All right, so to fill you in on what the virus replication program looks like, okay? So the image on the right there, it takes you through each of these steps, and I'll just animate through this to point out a few important factors. So Ebola virus, like all viruses, attaches to the cell surface through receptor interactions, and that's shown at the top there. And these are through sort of more or less sort of non-specific sort of interactions, but like interacting with phosphatidylserine or having the cell sort of bind sugars on the virus particle, as well as maybe some more specific receptors like HER2. And then what happens is the cell is triggered to reach out and grab the virus through a process called macropinocytosis. That's what's represented by that arm extending around the virus particle. Then the next step is the virus is taken up into the cell through a process called endocytosis, which is basically the cell swallows what has been sort of grabbed by the macropinocytic step. And then there are proteases, cathepsins, represented by CATBL, that cleave the glycoprotein. The glycoprotein that can then, which is the cell surface, the virus surface protein. The glycoprotein then binds a protein called Niemann-Pick C1 protein, which is sort of an internal endosomal receptor for the virus particle. Then the virus triggers calcium flux into the endosome through a protein called TPC2. And then through endosomal acidification, the virus glycoprotein undergoes additional conformational changes which cause the particle to fuse to the endosomal membrane and be released into the cell cytoplasm. So once inside the cell cytoplasm, the viral genome, which is negative strand RNA, single-stranded negative strand RNA genome, is transcribed by the RNA-dependent RNA polymerase into positive strand genome as well as virus mRNAs. And the mRNAs are transcribed in a gradient with the most on the left side of the genome and the least on the right side of the genome. And so there's sort of a gene dosage effect that occurs because of that. And two of the proteins I want to point out here, one is the nucleocapsid protein, NP, and another one is BP35. They are two of the most abundantly transcribed and in the end translated proteins. So once the proteins are being made, new viruses assemble in what are called inclusion bodies, and then they are trafficked to the cell surface and released. So the important thing for this talk is that each step involves many host factors and proteins, and they're shown by these shapes here that are different colors just appeared. And each step is carefully timed. It's much like a computer program. So if you take a computer program and swap all the instructions around, you'll just get, nonsense. Nothing will be produced. The same thing for a virus. If you disrupt the virus's program in any way, change around the sequence of the programming steps, or even the timing, you'll get nonsense. You won't get any virus being produced. At least that's the idea. And that's what we wanted to look at in what we did here in the study. All right, so how do we identify the host factors that are controlling the timing of the virus infection program? So what people did previously was they did a number of different whole genome screens, whole genome screens or partial genome screens to identify these host proteins. All right. Now, the ones that, and they're listed here. If you just focus on the output of these screens, you'll see I've listed several of these as being CPE survival, that's in red. And there's a couple of screens that look at RNA replication. All right. Now, what is CPE? So CPE is a so-called cytopathic effect. So when a cell is infected with a virus, and shown on the left here with the little cartoon, the cell gets infected, the virus sets up shop and disrupts the normal cellular function. This causes the cell to eventually die. Okay, and that's the cytopathic effect. So what these screens all did was they used that effect to select for cells that were perturbed, so the virus program was disrupted, and therefore the cells would survive. So no CPE took place, right? That's a selective strategy. So the thing is, what happens when you use CPE as the outcome? It's very sort of 0 or 1 type type of outcome, right? If you get even the tiniest little bit of infection, the cell will still die and have, the CPE effect, the cytopathic effect. So what generally happens in this type of screen when you're using CPE is you identify host factors that are very strict in the way they're controlling infection. So for instance, NPC1, if you don't have NPC1 expressed in the cell, cells do not become infected virtually at all, right? And so that's acting at the point of cell entry, all right? So, and what you see here, if you look down the main host factors that were identified, you see factors that are all involved from these CPE screens, all involved in uptake into the cell and getting out of the endosome into the cell, into the cytoplasm. There's very little involved in the next steps of infection process. So you're focusing on entry factors, typically, generally speaking. All right. So one of the goals that I wanted to get into more, well, and when you're manipulating cell entry, right, you only need one virus to get past that cell entry block, and then replication will start, and you can't stop it. So what I wanted to do was try to do a screen that gets past identifying entry factors and get into more of the weeds of the replication process to identify things that I predict are going to be more amenable to therapy development. So two screens that attempted to do this, one was, they're listed in green, by Cheng and Martin et al. They used different approaches to figure out, to identify replication. The one by Martin was the one that was done most thoroughly, and indeed identified replication factors CAD, NXF1, DDX39. So we wanted to build on something like the Martin study to identify additional factors that could be used as therapy targets. So what we thought about is, all right, how can we do the things that we do well in the BSL-4 that are most amenable to working in the BSL-4, and what my lab does best? And so if you remember, if we treat material in formaldehyde to fix them, we can take it out of the lab. And also my lab is very good at microscopy, right? So we wanted to marry those two things together to do a large screen. And there was a paper published by Nambo et al. In 2013 that helped us to figure out the best, a good approach that could potentially be used. All right. So if you look at what they did, was they looked at virus protein expression patterning over time. And you can see, so say, if we just look at the left set of images, it says negative sense RNA at the top and NP expression. So they used what's called a fish stain to identify virus RNA, and that's shown as the green staining pattern. And then they used antibody detection of this virus protein called nucleocapsid, and you can see that stained with red. All right, and the important part about this is that over time, it's not just simply the amount of protein and RNA that's being expressed. It's how it is expressed and the type of patterns that it forms. And that, what I call that a phenotype. Okay, so you see here, NP starts out being as little dots. These little dots get larger and become sort of more as sort of circles. And then these fill out with nucleocapsid protein, and then the circles sort of disperse and become more general through the cell, and then eventually, as the viruses leave the cell, it becomes much fainter and even more dispersed. And if you look carefully, you'll see it extended to the limits of the cell body. The same thing applies to the positive RNA on the right there, but it's not so important. The thing is, though, that's represented by this animation on the right, with the timer, is that what you're seeing here is you're seeing a virus program in action, right? So there are proteins coming in, virus proteins and cell proteins coming in and out of complexes, and this drives, it acts essentially as a timer, which drives the process forward. So we want to take advantage of this. So we thought to ourselves, maybe NP is not the best choice, though, because it's the most strongly expressed virus protein, if you remember back from to the genome part of the work. So we looked at a protein called VP35. VP35 we thought was a better candidate because it's a multifunctional protein. It was well characterized by Chris Basler, Guy Ramasinga, Daisy Leung, and myself. We've been doing collaborative work for many years. It also does some interesting things like inhibits the innate immune response, alters the way the cell detects virus RNA inside the cell body, and also drives the switching between positive and negative virus genome RNA synthesis. And importantly, we had very good monoclonal antibodies that could be used in a type of screen. Lastly, from Nambo's paper, it showed a similar patterning as you'd see with NP. So we thought this was a good choice. All right, so Callie Donoghue in my lab, a graduate student who's now at the NIH, she decided to see this in action for herself. And indeed, she was able to show this one's NP and virus RNA, but the same thing applies to VP35. She saw a patterning of infection that occurred over time, and the cartoons below show what's going on in terms of virus replication over time. But the images at the top show how the patterning changes. So you can see NP starts as these little round dots with the virus RNA sort of being bound throughout the cytoplasm. Over time, the little round NP dots turn into larger sort of pools of NP. And if you look at the dot on the lower right, it's filling it up with virus RNA. We believe this is where the viruses are being formed. And then it turns into this sort of what I call sort of a supernova type of look, where the NP becomes more dispersed. And then the RNA seems to form these sort of pools through the cytoplasm. And eventually at late replication, you see the viruses leave the cell and the RNA is depleted as it fills up capsids. All right, so the big deal is, how do we actually work this into a screen, right? So, the sequencing should look like this, right? And we get information about what virus proteins could come in and out of complexes. But unfortunately, what people generally get is they get a muddle of everything, all mixed up together. All right, so how can we pull this apart to get at the virus, to get at what I call the time map of the virus replication process? And the thing is that, if we knock out a host factor that, say, controls step C, shown there with the red block in it, right? If we knock out the host factor that's controlling that step, we should see accumulation of step B and a loss of step D, right? And that's sort of exemplified in this image on the right there, where you can actually see groups of cells that have been manipulated to block different steps. And you see accumulation, for instance, of step B and a lesser of step D. Right. So how can we harness this type of effect? All right, so what we did was we used a process called optical pooled screening. And this was developed by Paul Blaney at the Broad Institute. And he published a paper in Cell in 2019. And they've been developing this, this technique over the years since. But essentially what you do is you take a CRISPR library in an lentivirus vector, put this into cells, and so you'll get a pool of cells that are now knocked out for each one of the CRISPR targets. And we challenge with Ebola virus, shown here by the squiggle, and then wait for 36 hours. Now the key is that what Paul could do was he could use a sort of a hack on the Illumina sequencing reaction and sequence out the particular CRISPR guide, the barcode for that guide, in every individual cell, as shown in this animation on the right there. So you can see the sequencing through the Illumina reaction is sequencing out barcodes for each CRISPR. So what you get is you get a measure of what CRISPR is in that particular cell. And then if you use on the lower, what's indicated on the lower right there, the time map that I showed you before, you can then look for accumulation of the 12-hour time point and a loss of the 20-hour time point. And that would indicate that particular host factor that's being targeted by that particular CRISPR, right, that we've sequenced, is controlling the part in the middle, which is something to do with the replication step, which is what we were mostly interested in. And we said, all right, let's combine this between virus RNA expression, because they have information about what the virus is doing in terms of making RNA, and virus protein expression, because RNA should give protein, and the protein also, as I said before, this protein is multifunctional and can tell you a lot about how the virus is manipulating the cell and what the virus needs from the cell to control its replication cycle. So we put these things together, and we did the screen. All right. So the screen was 4 guides per 20,000 genes. All right. So we're knocking each of the host's 20,000 genes out using 4 possible CRISPR guides. All right. We are using trying to get to a point where we have at least 150 cells per CRISPR guide, because that gives us enough statistical power to drive the screen. And if you do the math, what we ended up with is 4 million 10 times magnification images. And we, like I said, we try to get above 150 cells per CRISPR guide. So we got to about 1,000 cells. Unfortunately, this equaled about 12 terabytes of image data, which is obviously an awful lot. But just as a sanity check, if we knock out MPC1, which is that critical endosomal receptor for Ebola virus, you see here a great loss in infection. And that's exemplified in this area under the curve plot on the right there, where you see a shift from the purple, which is the non-targeting CRISPR, to the left, which is the MPC1 knockout. So the screen is working when we can get good statistical data out of it. Right, so like I said, 12 terabytes of image data is just too much. So we worked with George Stephanakis in Carolyn Uhler's lab at MIT, and he developed a machine learning model that's sort of known as AI these days, to automatically identify the different phenotypes that are shown with those black images just below this sort of the diagonal sort of matrix in the middle left there. So the AI was able to identify faintly expressed punctate expressing cells for BP35, the so-called cytoplasmic diffuse staining pattern, and then a peripheral state where the cells were probably, the virus is leaving the cell. And this procedure worked pretty well, and was able to show that we could differentiate between MPC1 lacking expression pattern, MP1 lacking cells with loss of infection, away from some other factors that were maybe restriction factors, shown on the right there. So just what happened in this. So we use one parameter model, which is just a simple, let's look at the difference between highly expressing cells, if you look down below the x-axis on this graph, versus faintly expressing cells. What we get is the usual suspects. We get entry factors, say MPC1, RAB7, SPNS1, all known from previous studies, those other studies I showed you before. And we get some factors that control, maybe restriction factors, like integrins, which are also known before, that may be holding back virus infection. So they become more intensely stained than you would expect from the norm. Right, and as you can look on the right there, you can see, factors controlling entry are predominantly shown in this list of Go terms. Now, the more interesting thing is when we combined two parameters. So if we look at the cells that are punctate staining on this y-axis on this graph here, versus those that are faintly stained on the x-axis, you'll see now we're getting division between those controlling cell entry, so NPC1 in that red circle on the right there, RAB7, SPNS1, away from things that are to do with virus replication. So many of these are pyrimidine or purine synthesis controlling enzymes. So that's actually makes sense. If you block the cell's ability to make RNA precursors, the virus is probably not going to do so well because it has an RNA genome. So indeed, this demonstrated the procedure worked quite well. And this is the first time that any such screen was able to differentiate entry away from replication. Some other interesting things came out of this screen was RNA stability, looking at the levels of virus RNA in the cell. And as you'd predict, this was identifying factors controlling RNA stability. So PABPC1 is a factor that we've been looking at more recently and has been discussed. It's a cell protein that binds poly A tails, controls mRNA stability. Two other proteins, TRMT61 and TRMT6, are actually part of a single complex, and I'll show you what that is shortly. And two other proteins, STRAP and SRPK1. SRPK1, and these are also part of a complex controlling the spliceosome. SRPK1 was previously described in literature as controlling Ebola virus RNA stability, so that was a great sanity check. And then STRAP was something unusual that we followed up in, and I'll show you a bit about that shortly. So TRMT6 and 61A, they're tRNA methyltransferases. So these control tRNA stability, but the important thing is these proteins also methylate the GU-CRA motif that's outside of tRNAs, and possibly virus genomes. So we think this pathway is somehow controlling RNA stability, and we're following up on that still. So an interesting thing was this protein STRAP. So SRPK1, like I said, was shown to be important in spliceosome regulation and was shown to be important for virus RNA stability, replication for Ebola virus. Now, STRAP is related to that particular complex. It positions the spliceosome in cells. It's not actually the spliceosome itself. It's not an enzymatic component, but it positions that structure within cells. And the interesting thing is what we were seeing was that even though there was more virus RNA, so more RNA, as shown by this AUC plot here, there was actually less virus protein, as shown on the plot on the right. And of course, RNA usually gives you protein, right? So more RNA usually gives you more protein. Whereas this is showing some sort of dysfunction, some sort of dysregulation of the RNA synthesis or stability of the virus. And we wanted to dig into that a little bit further. So what we did was we did some more quantitative analysis, and we found that, if we do qRT-PCR, we indeed find that the RNA is up inside the cells. So that confirmed that area under the curve plot I showed you before from the fish analysis, but this is using qRT-PCR, so it's more quantitative. But what we saw was that the virus RNA in the cell supernatant, which is where you expect virus to be, right, was down by up to around 10-fold. So that's interesting. So more virus RNA in the cell, less virus RNA in the cell supernatant. And if you actually look to see, what productive virus in the cell supernatant, you see that dropping by about 5-fold, right? So that's directly proportional. We've lost virus RNA in the supernatant. We have less virus, infectious virus in the cell supernatant, but it actually not shown here on Western blots is the amount of virus protein was sort of the same. So we're producing what seems to be defective virus particles. And if you look on this plot in the middle here, it just shows virus spreading from cell to cell. So the cartoon up the top shows how virus infecting in the middle spreads from cell to cell. You see a lot, the foci of infection are much smaller than normal. And if we look to see where strap is expressed in the cell, you see green puncture where strap is, and then VP35, which is the protein we're looking at in the screen, overlaps almost perfectly with those green puncture from the strap. So the two proteins associate in some way. Probably the virus protein is disrupting strap function, but strap is somehow important for virus RNA stability. And if you remove it from the cell, you then have a defect in the replication cycle. Right, so just to wrap up, so this screen was productive in identifying many new factors that had remained unknown from previous studies. So we identified 946 new factors over the 270 from previous mass spectrometry analyses. We identified new viral RNA stability factors, and you know, these new networks, these new factors can be put into networks to better understand the replication cycle. So overall, the optical pooled screening was highly successful in identifying host factors involved in replication. And using this sort of phenotypic readout by microscopy, I think, is very amenable to you know, other looking at other pathogens inside cells. Some problems with the approach: it produces a lot of images, and to analyze these effectively, you need a lot of computational power. And you know, of course, computational power is improving, you know, doubling every year still. And AI tools will make this even more effective in the future. You know, and the way we did the screen definitely can be developed and improved. Right, so some next steps: we intend to add in extra markers to try and look at the infection cycle with more detail, and this is ongoing work in my lab. So I'll finish up. So just to acknowledge Paul Blaney, of course, who developed the optical pooled screening system, and Rebecca was in his lab working with Justin Patton from my lab. Carolyn Uhler was supervisor for George, who developed the machine learning tool. And my other collaborators, Daisy, Chris, Gaia, and members of their lab, they've helped. They helped to get this through to publication, a successful outcome. Of course, members of my lab mucking around their BSL-4 suits at the top there, and lower when we're ASV, again mucking around, having a good time. All right, so I'll stop there, and I'm happy to take any questions from the audience.
All right. Thank you so much, Robert, for your very informative presentation. We will now go ahead and move into the Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just click on the Ask a Question box located at the bottom of your screen. Okay, let's go ahead and get started. Okay, the first question that we have here says, were any of the host factors identified in your CRISPR screen already known, and did their timing of action surprise you?
Yeah, so that's a great question. So about, like I said, about 200, sorry, wait, about, gosh, I have to remind myself, about 28 of them were known just for the protein expression shown in this slide here. And then about 46 were known for the replication thing. So the thing that was most surprising was this involvement of strap. So we expected strap to be, you know, the loss of this factor to more or less cause a loss of virus RNA, and this would cause a loss of protein in the cell. So when we first analyzed this, we said, oh, look, we knock out strap and we're not getting infection. I bet you it's something to do with, you know, there's not as much virus genome inside the cell. And this equals not as much virus protein inside the cell, right? That's pretty straightforward. But actually it was upside down. There was actually more RNA in the cell and less, you know, less protein being produced. So we're going, huh, what does that really mean? So we're thinking it's actually, you know, when you, I think it's the jumbling up of the lines of the computer program, right? So when you jumble them up, when you knock out strap, you're starting to jumble up the lines, and this is causing sort of a dysfunction of what's being made when it needs to be made, right? It's no longer in a normal relationship, and that causes the virus to sort of self-destruct or leave the cell in an immature state, so it can't infect new cells. So that was, yeah, pretty interesting. And we see this a lot, actually. We see this quite a few times. It's just hard to understand it. You have to really think deeply about what's going on.
All right, great. Thank you so much. The next question that we have here, it says, my big picture question is, why has it been so difficult to find small molecules / drugs for Ebola and many other viruses, even though antibodies and vaccines have been effective?
Yeah, that's a great question. So one of the one of the issues is when people show up to the clinic, right? So people, you know, obviously you're not going to go to the hospital when you're feeling all right. and if you're infected with Ebola virus, it's a very sort of slow replicating virus. So, but you know, between when you're infected and when you start feeling ill, it's actually quite a long time. And because of that, there's a lot of virus in your system. So if you go to the clinic now and you get treated, it's really hard for those small molecules to do their work because there's so much virus in your system, right? So what you need to do then is to try to find treatments that are working very late in the infection process, and that's just really incredibly hard to do. So I mean that's really the main barrier, right? So we have a lot of things that work in the lab in the plastic plate, right? We have cells, we treat with the virus an hour later, and then we treat with the drug, right? That's the easy bit. But actually getting it to the clinic is the really hard bit, and that's where we need to, you know, really have very, very strong treatments to do that. And we're getting there. It's happening, but it just takes a long time.
All right, fantastic. The next question that we have here says, what challenges did you face in implementing large-scale CRISPR and imaging screens under high biocontainment conditions?
Yeah, sure. The main challenge is, you know, like I said at the start, we can only do certain things inside the BSL-4 lab. We're limited by time that we can spend in the lab, and also we're limited by the way we can remove stuff from the lab in a safe way, right? So we have to think very strategically about what we're doing. So for instance, you know, we can't do a really great time course of infection because it means that somebody has to go in there at midnight, which is just difficult to do. Whereas you could, you know, in a BSL-2 lab, you can just walk in and off you go. Whereas in a BSL-4 lab, you need to have somebody on the radio, you need to have a buddy who's watching, you know, making sure you're okay in the lab. You know, it's those things, it's those, you know, the sort of boring things that sort of slow us down. But we try to work out the best ways to be efficient and approach it like that. And I think this particular screen was a good middle ground to get a lot of good data, but to do it safely and to do it efficiently as well as we could.
Right, great. Next question here says, could this screening approach be adapted to study other high consequence viruses, and what might that reveal?
Yeah, so I think it could certainly be applied to any type of virus. So you just need to have a robust way of detecting the infection process. So, fish staining, but looking at RNA viruses is very effective. I think in our study, it could be improved to make it even more, detecting more details and making it more granular. And that's what we're looking at next. But having really great antibodies. So Daisy Leung's lab gave us a really great VP35 antibody, and that's what made this thing work well. So just make sure that your staining process and your detection of the infection is strong and very uniform, and then you're set. If you don't have that, then it's much harder to achieve a good outcome.
All right, great. We will go ahead and wrap up with this final question here. This one says, how does resolving the timing of Ebola virus infection change the way we approach antiviral drug development?
Yeah. So, like I said, a lot of the screens beforehand had been looking at entry factors, right? And the thing with controlling virus entry is you really have to it has to be complete, right? If one virus particle sneaks past that treatment, the cell will become infected, right? So that's a high bar to reach, but still achievable. I mean, there are still, excellent drugs out there that could block cell entry, and that's still a great goal. I think, combining those type of drugs, small molecules, with something that's controlling the replication process. So if that virus sneaks through and starts to replicate in the cell, you can then have another small molecule that interferes with that step as well. So this particular study, right, was trying to identify those things that could complement something blocking cell entry. So combining the two, you should have a pretty fail-safe way of controlling the infection process in patients. So that's the objective, and that's what I think we got very great data to inform, and then to design drugs around those particular targets. That's what we're doing next.
Perfect. Okay. Well, thank you so much again, Robert, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand for 1 year until September 3rd, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thanks so much, everybody. Goodbye.